I won't repeat the title because it's very long, but that's what I've been working on lately a lot. And this is something that was initiated, and most of the data were acquired before I actually left Jesper's lab at the end of August. So now I'm working actually at the Department of Surgical Sciences at Uppsala, but since the project is a project from Copenhagen, so you would still see the nice Olsen Group PowerPoint frame. So about type 1 diabetes, of course, many are familiar with it, and what it does is it leads to the destruction of beta cells that produce insulin. So then you can't produce insulin anymore, and then you can't control your glucose level. The patients are relying on daily injections of insulin for life. It can require pancreatic transplants in very bad cases, and there's no cure so far. So of course, there's high interest in curing this type of disease. And here's the architecture of the pancreas, and what you can see here is that actually that's on this particular microscopy picture, that the islets don't represent a large portion of the mass of the pancreas, and they are so they are scattered in the pancreas, and they are composed of beta cells, alpha cell, beta cell, and some other less common cell types, and at different percentages, of course. And this is how we actually get our islets for single cell sorting. So it's actually a protocol that's been developed for islet transplantation. So it starts with perfusing the pancreas with the collagenase, and then incubating the recorded chamber, produce like this pancreatic digest, and then by centrifugation you can isolate the islets, and then you get this pre-purified islets. And then usually they will go to transplant, and they will be injected in the vein, and then they will settle in the pancreas. But in our case, we stopped before, and we analyzed these islets. And this is actually a picture of the purification that was done. So this is not something that I'm doing. It's actually done by our collaborators at Uppsala, so Dr. Louise Granlund and Professor Ole Korsgren, who are specialists in pancreatic transplant and diabetes. And here you see that at least for this particular isolation, there was very few contamination by exocrine tissue in yellow, and you mostly see islets, so it was very pure. And we actually sorted these islets and did the proteomics analysis on it. So of course, that was all done at Copenhagen. So they were transporting the islets, and they have a protocol to actually transport these islets and still keep them and be able to transplant them from one country to another. So the islets we got were also transplantable grade islets, so the highest quality. And then for the stem cell derived islets, we collaborated with Chen-Cheng Wang and Professor Hanne Scholz from the University of Oslo. And what they did is so create the stem cell derived islets. Then they brought them back for proteomics analysis in Copenhagen. And then Chen-Cheng went back to Oslo and then transplanted them into mice. So it was not in pancreas, it was in the kidney capsule of mice. So they are not influenced by the pancreatic environment, but they will be influenced by the glucose level in the blood, and then they will mature in the mice. So the goal was to do then analysis in December, which actually is still ongoing at Copenhagen at the moment, and it's done by Michael Lechner, if you have heard of him. So basically we did the analysis before the maturation and after the maturation. It's important because these islets, we're not sure to which point they are mature before the transplantation, and then that will have an influence in the treatment in vivo. And this is the workflow we've been using for quite some time now for single cell proteomics. So we do the sorting with the cellenONE into this Evo 96 proteochip, then transfer to the Evotips, to the Evosep system, and then injection into Astral. And here, I think Nikolai's word resonates a lot with what we've been doing. So the important part actually for this project was really the Whisper Zoom. because so we wanted to initiate the project earlier, and Louise and Ole were putting a lot of pressure actually to start early, but I told them to actually wait because we knew that the faster version was coming, so I didn't want to do the analysis with 40 SPD, and hopefully we could have analyzed twice as many cells per sample and get like better insight. So what we did when we got actually the WhisperZoom for testing is we did the tests we had to do, which were probably for 3 or 4 days, maybe 5, and then right after, the week after, we were analyzing sample. And why it was important is because type 1 diabetes pancreas, they rarely get them at Uppsala. So they got one last year, and that was the one the week after we installed the WhisperZoom. And that's the only one they've had since then. So if we didn't do this analysis on this type 1 diabetes donor pancreas, we would not have been able to do that. And that's pretty much that has been completely enabled by the WhisperZoom. Of course, all this analysis has been done at 80 SPD with the WhisperZoom. So starting with the pancreatic islets from healthy donor. So first thing I did, of course, is check the level of hormones, pro-hormones that you can see in the islets. So this is around 400 cells with a depth of more than 4,000 proteins, and on average slightly less than 4,000 protein. And what you can see is you have a lot, of course, of glucagon. Much less of somatostatin expressed, and a lot of insulin, which matches pretty much the proportion of the cell that we're expecting. So beta cell with 60–70%. So here it looks more like around 50, and then less of the alpha cells, delta cells around 10%, and then the cells expressing pancreatic polypeptide, not that many, and ghrelin not that many either. So it matched the population very well. And what we also saw, and this has been actually reported recently, not published, but observed by other groups, that some of these cells express multiple hormones. So we're thinking about looking into that later. So we already had some interesting observation just looking at the healthy islets. Now, what I did first also is look at markers, of course, and what better to do than looking at single-cell RNA-seq atlases, which have been very popular, and they are easy to access online. And so I picked up some markers, for instance, in that case for acinar cells, and trying to test to see how it looks in our data set. So here just the healthy donor, the cells from a healthy donor that are plotted PCA, and the scaled abundance of the markers. So you see that seemingly a lot of these cells have the expressed marker that should be acinar cells. But if you look at the insulin abundance, they actually, some of these cells express both markers, which shouldn't be the case because insulin cells, producing cells are endocrine cells and they are not acinar cells, so you shouldn't have these markers. So looking at, so that's how it looked, for instance, in the article for the single cell RNA-seq. So the blue, the cell with in blue in the cluster are acinar cells, and the cells that are just on the left and a bit below are the endocrine cells. And you see that the endocrine cell have absolutely no expression of the mRNA of this particular gene. But what we see is that actually it doesn't seem super specific, at least at the protein level. And looking at another marker that this study reference, you see many less cells that have that express it, and they are likely to be actually the acinar cell. And if you do a correlation, they didn't correlate very well. What it looks like is that this population was actually probably the acinar cells. And here you also see that thankfully don't correlate so well, but it looks like there's an anti-correlation a bit with at least with the marker that we think is more specific, an anti-correlation with insulin. So now looking at healthy donor, type 1 diabetes donor, and stem cell-derived islets. And so this is how it looks for more than 1,000 cells, so around 400 per, tiny bit less than 400 per condition. And here I just highlighted markers that I picked up from either same atlases or the literature. And what you see in gray is missing values. So what you can appreciate first, so continuing on acinar cell, is that they were very present, for instance, in this type 1 diabetes donor islets. And The reason is because for that particular one, we knew that there will be a lot of acinar cells because the purification, the preparation was less pure. So that's something we're expecting. And you see, for instance, this the marker that I was highlighting before, which is actually expressed in all the cells. And then this ISK1 that is a bit more specific. But what you also see is the actual enzymes that are secreted by these cells, like for instance, alpha amylase, sorry, pancreatic alpha amylase, and colipase, which are enzymes that are secreted by acinar cells. And you see that you have high level of those and not so many in the rest of the cells. Actually, almost zero, just a residual expression. And then for the iPSCs, where we expect absolutely zero acinar cells because they were not differentiated to make acinar cells, you see absolutely zero expression of these enzymes. Meaning that that's probably better markers, and then we can actually confirm the fact that these iPSCs made the endocrine cell they were supposed to make. And here's the correlation, for example, for two markers like the pancreatic alpha amylase and the colipase, and you have like a pretty good correlation compared to the other markers that we were looking at before. So what we see also looking at the stem cell is that they somehow expressed high level of every of the hormones, every hormone. So for instance, glucagon here, you have insulin also that is highly expressed in every cell, and you have a high level of somatostatin, which seem to indicate that they are not very oriented toward a particular type, but this seems to be still a bit immature. And that's also highlighted with other markers such as chromogranin A and PAC6, which are, so PAC6 is expressed during embryogenesis a lot in the fetal pancreas, and then chromogranin A also, so indicating that these stem cell derived islets are immature at least before transplantation. Of course, what we could see is that in the type 1 diabetes patient, there was not many cells expressing insulin, which is of course expected, but it's also expected that there are some that still maintain expression of insulin, and it's been also documented. And here are actually the level of each hormone. So here insulin, you see strikingly like a group of cells in the type 1 diabetes patient that express insulin at a similar level as a healthy donor. So they probably have actually functional very few of these beta cells, which is again something that is that's been described. And then here's the expression for on the right for glucagon, which they are then in that case similar level in type 1 diabetes and the healthy donors. Makes sense because these ones are not necessarily lost during the onset of the disease. And finally, you also observe somatostatin, and for pancreatic polypeptide, we don't see it in stem cell, which is nice because they were supposed to make enriched for B cells, for beta cells, sorry. So they were not supposed to express this type of markers. And what you can also appreciate is somehow the markers, the hormones are expression is always a bit higher in also stem cell, and they seem to express at least these three at high level. And finally, we also looked at other key marker quickly. So in that case, TNF-α, which is indication of inflammation, and it's also been shown that anti-TNF-α treatment slows down the type 1 diabetes progression, but very little, and it doesn't treat, of course, the disease, but we see higher level of TNF-α, for instance, in type 1 diabetes cells, which match very well with observations. And here is, for instance, chromogranin A that I was mentioning before. It was extremely high in stem cell derived islets, and this is also, for instance, a marker that is used for neuroendocrine tumors and cancer. So it's something that is expressed in the endocrine cell in the pancreas, but not at that high level, so meaning that the cells are likely to be still a bit immature. So then sadly, I don't have the analysis yet on the cells that have been matured in vivo, but the expectations are that these we will be able to see like the proper phenotype of the cell, so the beta, the alpha, and the delta, and then all these markers that indicate less maturation should fade away. And here, in that case, if you, for instance, if you check like the correlation of, for instance, insulin against somatostatin. You have no correlation for healthy donor, but for stem cell derived, you had like a positive correlation, which was not great, but still there was indication that higher level of insulin, cells with higher level of insulin at higher level of somatostatin, which is not something that you want to have. And the take-home message is that single-cell proteomic analysis is now able to highlight these differences at a very good depth. So we had up to more than 4,000 protein quantified per cell, and they are smaller than, for instance, HeLa cells. And we can identify some of the cell types without using so far complicated classification. And that was also shown actually in an article that came out recently at Lower Depth by James Fulcher. And if you're interested, you should check it out. But they also actually did the splitting to do RNA-seq on it, so it's a very cool method. And then what we saw is that the stem cell derived islet are not fully matured prior to transplantation, but they show a complete absence of some of the cells that we didn't want to see, for instance, acinar cells or gamma cells. And yeah, the bottom line is to correlate to collaborate with with biologists, because now that we have all the workflow that seems to be working, and it was very robust. So as you may appreciate, that's a lot of MS times, and it's been very stable. So we are able to collect like this type of big data set. And of course, the Whisper Zoom has been a component of the data set. So sadly, we didn't do any method development, so it just worked and we used it, and that's what I think it's been made for. So we truly appreciate it to be able to access it early and be able to record this data. And with that, I want to acknowledge quite a lot of people from, of course, the whole Olsen group, and particularly Jesper and Maiko, Louise and Ole from Uppsala University, Chen and Hane from Oslo, and then the Evosep team, particularly Ole, Nikolai and Dorte, and also Anjali and Fabiana from CNION, and yeah, we got help from Diagnosis at some point, so, and the whole Evosep team, of course, and thank you for your attention.